Hi, this is Remembering the Past, the show where we talk about people who've died recently who've had a profound effect on our history, our society, or our culture. And in our ongoing series on Nobel Prize winners, tonight we're going to talk about Sir John Sulston, who died recently at the age of 75. Dr. Sulston was awarded one-third of the 2002 Nobel Prize in Physiology or Medicine, along with Sidney Brenner and Robert Horvitz, for their work on the genetic mapping in the organism C. elegans, which is a nematode, a roundworm, and this work led to their discovery of a gene that is responsible for programmed cell death, which is now a critical concept in biology. This was truly a team effort and a multidisciplinary effort. The work took place in Dr. Brenner's laboratory, where he was the one who decided to use a nematode roundworm because of its cellular simplicity. It's only composed of about 1,000 cells, and yet its functional complexity. This made it the ideal candidate to study the form and function of its genome. Dr. Sulston, whose early background was in organic chemistry, was the one who actually mapped out the genome in a painstaking process almost by hand, and Dr. Horvitz was the one who isolated the gene that's responsible for programmed cell death, which is also known by the scientific term apoptosis. I just want to put a plug in for Dr. Horvitz, by the way. He's a nice Jewish boy from Skokie. He actually went to the same grammar school that I went to in 7th grade, East Prairie, and he went to Niles East High School, which was my sister high school. I actually would have gone there had I not moved. He is one of 2 Nobel Prize winners from Niles East High School, the other being Martin Chalfie. There are very few schools in the world that can claim 2 Nobel Prize winners. And let's face it, that's as close as I'm going to get to any Nobel Prize winners. Anyway, back to John Sulston. He was born during the war in Cambridge to an Anglican priest and his wife. He had poor eyesight and was a miserable cricket player and wasn't interested in much besides the sciences, especially biology. And he went to Cambridge in 1960 to read in the natural sciences at Pembroke. He was losing interest in the biological sciences and actually getting interested in theater lighting. And probably one thing that changed his life as much as anything was he liked to party, and he was in a drunken revelry one night and had to be bailed out by one of his tutors. And that sort of changed his thinking about what he should do. And so he went to organic chemistry, where his job prospects would be better. One of the things he talked about later in life was the intersectionality between the sciences, which benefited his career. And here he talks about his beginnings in organic chemistry.
The benefit is enormous, and I find it easiest to understand because I'm not a human geneticist, strangely enough. In fact, I'm an organic chemist. I'm an absolute bottom of the sort of pile in terms of biology, really. I'm an organic chemist, but I understood from the way the nematode sequence worked how valuable it was that as soon as you start to put gene sequences into the hands of geneticists, even though they don't even know which gene they're looking at to start with, you give them all the sequences, they can hunt around and look for the one they want and then work on it and understand it.
Sulston actually got into genetics in a roundabout way. He was recommended for a postdoctoral at the Salk Institute in California, and his mentor there introduced him to Sir Francis Crick of Crick and Watson fame. Crick interviewed him on behalf of Sidney Brenner, who was working back in Cambridge, and he ultimately went to work in Brenner's lab, working on the C. elegans and mapping its genetic makeup. While he was doing that, he ultimately discovered a gene that eventually was found to program for cell death in the roundworm. Let me do a brief explanation of why programmed cell death is important. For any organism to survive, certain cells must thrive for a while and then ultimately die, and they are genetically programmed to die, which sounds counterintuitive, but it's not. When I learned about this, the best example I found was the deciduous tree, trees that grow new leaves every spring, but to do so, they have to shed their leaves in the fall. That autumn shedding of leaves, or dying of leaves as it were, is programmed into the tree every year. And it's like that for other organisms too. So tadpoles lose certain structures as they mature to become frogs. Caterpillars can only turn into butterflies after certain cell deaths, and it happens with humans too. Here at a Nobel press conference, they describe cell death and the work of Dr. Sulston.
The human body consists of hundreds of cell types, all originating from the fertilized egg. During the embryonic and fetal periods, the number of cells increase dramatically. The cells mature and become specialized to form the various tissues and organs of the body. Large numbers of cells are formed also in the adult body. In parallel with this generation of new cells, cell death is a normal process, both in the fetus and adult. To maintain the appropriate number of cells in the tissues and for proper morphology of these tissues. Delicate, controlled elimination of cells is called programmed cell death. This year's Nobel laureates in physiology or medicine have made seminal discoveries concerning the genetic regulation of organ development and programmed cell death. Possibilities were opened to follow cell division and differentiation from the fertilized egg to the adult. The laureates have identified key genes regulating organ development and programmed cell death, and have shown that corresponding genes exist in higher species, including man. Knowledge of programmed cell death has helped us to understand the mechanisms by which some viruses and bacteria invade our cells. We also know that in AIDS, neurodegenerative diseases, stroke, and myocardial infarction, cells are lost as a result of excessive cell death. Other diseases like autoimmune conditions and cancer are characterized by a reduction in cell death, leading to the survival of cells normally destined to die. John Sulston extended Brenner's work with C. elegans and developed techniques to study all cell divisions in the nematode, from the fertilized egg to the 959 cells in the adult organism. In a publication from 1976, Sulston described the cell lineage for a part of the developing nervous system. He showed that the cell lineage is invariant. That is, every nematode underwent exactly the same program of cell division and differentiation. As a result of these findings, Sulston made the seminal discovery that specific cells in the cell lineage always die through programmed cell death, and that this could be monitored in the living organism. He described the visible steps in the cellular death process and demonstrated the first mutations of genes participating in programmed cell death, including the so-called NAC1 gene. Sulston also showed that the protein encoded by the NAC1 gene is required for degradation of the DNA of the dead cell.
Here's the presentation of the 2002 Nobel Prize in Physiology or Medicine to Dr. Sulston in Stockholm.
Sidney Brenner, Robert Horvitz, and John Sulston. Your discoveries concerning genetic regulation of organ development and programmed cell death have truly opened up new avenues for biological and medical research. On behalf of the Nobel Assembly at the Karolinska Institute, I wish to convey to you our warmest congratulations, and I ask you to step forward to receive the Nobel Prize from the hands of His Majesty the King.
Dr. John Sulston.
Well, Dr. Sulston came from a religious background, and although he renounced his father's religion, he retained his father's sense of ethical scrutiny, which was especially important because much of his work was going on during the Human Genome Project, the Moonshot Project, to map the entire human genome, and so people all over the world were concerned with the ethical implications of what we might learn by mapping the genome.
The ethics is ourselves and our social systems, actually. I don't think inherently we're creating worse ethical problems with biology than we did certainly by, for example, inventing nuclear weapons. But in both cases, apply the democratic process properly. Of course, we do have to bear in mind all the time, as Winston Churchill said, that democracy is the most awful way of governing ourselves until you think about the alternatives. I think it's very true that we don't have any alternative to democracy. And unless we apply it properly, and that means a rather full democracy, not a microsecond response referendum, but a full democracy in which everybody has a chance to learn and to participate, then I think we may be in some trouble through, I don't know, discrimination of various kinds. And people have painted various horror stories, of how one might actually speciate ourselves if the rich sort of start breeding themselves in some clever way. I believe that properly applied social democracy will be a sufficient check on excesses of that kind.
Dr. Sulston took a broad view of his work and of science in general, and one of the things that he was especially emphatic about was the peer review process. He decried science by press release, which these days is even more prevalent than ever. You see people rushing today to get the results of their work into newspapers or into the media, and especially with the internet, Dr. Sulston thought this was not conducive to good science.
It's an old issue with science, I think, whether one is going to simply make claims. In fact, in a previous discussion, we were saying how the alchemists, I think, practiced science by press release, and it didn't do very well. The point about press releases is you don't check the facts behind, and what you should do with press releases is to say, show me your data. Now, biology is now finding itself in this position because suddenly biology is of commercial value, and there's a great temptation to try to gain a financial advantage by issuing statements but not publishing them properly. We've had some experience of this with genome work, and so that's why I made a particular point of this, that we must publish openly, show the data, and that's the way science proceeds properly.
Yeah, last comment may have been a veiled shot at Craig Ventnor, who was doing the private human genome work here in the United States. And Dr. Sulston, and this may be part of his English socialist background, was dead set against excessive profiteering from scientific research in the field of genetics.
Human gene, and you have sort of trails of discovery, i.e. you may find diagnostic tests that may be useful for a particular gene, or you may be embarked on a much longer, more tortuous and uncertain route towards maybe an eventual therapy. How are we going to deal with the intellectual property? Well, I think it's entirely reasonable for somebody who has a diagnostic test to put a barrier on that, to collect fees if anybody else wants to use that information. I don't think it's reasonable. In fact, I think it's really anti-competitive in a way which is not good science and not good business to allow them to put a block across all possible diagnostics. The current patent law allows you at the moment to write broad patents that not only cover that, but all of this on the basis of the very, very little information about the gene. This is wrong, it's not good, and it's not good for anybody, and we need to roll this back. But this is what's going on in biology today. These broad patents are being issued, and they will not serve us well in any way at all. And equally, we cannot expect to move forward unless we have much more public funding. We've got to accept the need for public funding. We cannot proceed to do most of the things that biology ought to be doing in the world, or this medical biology I'm referring to in particular, without public funding for neglected diseases. It's not a question of patents here, it's just a question of no markets. If you're working on malaria or TB, there is no market for a cure. If you're working on antidepressants, there's huge markets. And so lots of blockbuster antidepressants are produced and compete with each other. The marketplace alone is not enough to do what I believe we all want to do, which is to help everybody as equally as possible and not just make the rich richer at the expense of the poor. But just to sort of summarize, these are ways forward. It is moving forward. We can, I think, we can respect our common goods. We can do this. We can have narrow patents. We can put in safeguards. It's only going to come about through democracy. But I think in the end, I must confess as a scientist, I do just want to come back and say I think what we're really about is learning, is the progress of discovery. I think that really the most exciting thing for me is to have been a little bit part of this stage of human development where we're learning to understand our bodies as machines, as molecular machines. If I manage to make a little tiny contribution to that, that's great. And that's really what it's about. But I think we've got to be aware that our progress of discovery also has applications, and that those applications should be for the good of all and not just for the few.
Dr. Sulston, you made a great contribution. I'm going to close on that note. I want to thank my producer and IT genius, Sid Taps. Looking for a final song to pay tribute to Dr. Sulston, I had to decide whether to pick a final song on cell death or a song about worms. And Dr. Sulston did say once, when we understand the worm, we will understand life. I couldn't find very many songs on cell death, but there is one good one on worms. It's Glowworm. The song was made famous by the Mills Brothers, but rather than play that version, I'm going to play the instrumental version by the great virtuoso guitarist Chet Atkins, one of the best. So as a final tribute to Dr. Sulston, here's Chet Atkins with a little bit of Glowworm.